Very good morning to one all present here. My name is A. Melvina, Assistant Professor of Biotechnology, Sri Adi Sundanagiri Women's College, Kambam. Let me see about the topic single cell protein. So, what is single cell protein? The term single cell protein refers to the total protein extracted from the pure cultures of the microorganisms. Here, the microorganisms are yeast, algae, filamentous fungi, bacteria, and it can be used as a protein-rich food supplements by human and animals, and which is also known as a microbial protein. History of single cell protein, which is used to named as microbial proteins. In 1966, MIT professor Carol L. Wilson renamed it as single cell proteins. The transportation of food was common in the past, but with increase of population, energy crisis has been encountered. So there should an alternate food or protein sources to overcome the energy crisis. In 60s, idea that the dried cells of microorganisms can become an ultimate part to solve this problem thus gained research interests among the scientists and industries. So in the 20th centuries, the single cell protein technology for the production of protein rich contents from the microbes on the large scale was established. So this is the production of single cell protein. Here the following steps were involved. First step is selection of strains of microbes and substrate. The second step is fermentation. Third step is harvesting. Fourth step is post harvesting treatment. And the fifth step is processing of a single cell protein. First step, selection of a strain of microbe and substrate. So this is very crucial step. The microbe selector should not produce toxicity in its biomass, and it should not harmful for the consumer to consume, and its selected microbes should produce a large quantity of protein, and the substrate should be cheap, effective, and allow favorable growth and easy to isolate. The second step is fermentation. So the production of single cell protein takes place in a fermentation process. This is done by selected strains of microbes which are multiplied on suitable raw materials in technical cultivation process, dried to the growth of the culture, and the cell mass followed by a separation process. So the fermentation should be done under sterilized conditions. The controlled conditions are temperature, pressure, pH, humidity, etc. Usually the fermentation process takes place by fed batch fermentation. The next step is harvesting. Harvesting is nothing but the fermentation yields a large number of microbial colonies produced from a single cell. So these colonies are isolated from individual cells by the method of decantations. The next step is post-harvesting fermentation. So here, isolated microbial colonies are subjected to various differential techniques, for example, centrifugation, washing, drying, etc. So this is the entire processing of single cell protein. Here, the produced protein contain impurities in it, for example, carbohydrate, nucleic acids, lipid contents, etc. So the pure protein isolation can be done by disrupting the cell wall through crushing, crumbling, cycle of freezing or thawing, grinding, and thermal shocks. So here the nucleic acid can be removed by the chemical treatment of a 10% of sodium chloride and sodium hydroxide treatments, and some thermal shocks will also given. So the enzyme treatments of the ribonuclease were given. So this is the entire processing of a single cell protein. Thank you.